right, fantastic, everyone. Welcome, and I hope it's not too hot or it's not too wet or stormy. We're glad that you are going to listen to our um, it's the National Advisory Neurological Disorders and Stroke Council top priorities next working group, and it's our sixth meeting. And so my name is Dave Jett. I'm the director of the Office of Neuroexposome and Toxicology here at the National Institute of Neurological Disorders, Stroke, or NINDS. We're going to have fun and we're going to talk about the neuroexposome, NINDS strategic planning, and the neuroexposome top priorities working group presentation. It's going to be led by Dr. Pam Lyon and Dr. Gary Miller, and then we're going to have some time for questions and comments. So the first thing I'll do is a quick introduction about the neuroexposome. So one thing I always say is that, as we go through this webinar, I'd like you to think of why exposomic factors could be the primary cause of some neurological disorders. So here, we're just showing that we know that genetics has provided a very, very good insight into the etiologies of a lot of most different diseases. But actually, when you look at it, most health risk factors cannot be explained by genetics just alone. And so a while back in 2005, Dr. Wild coined this thing called the exposome. And now sort of all inheritable factors affecting gene expression are what we're calling the exposome. And so I think this is a new frontier of biomedical research that will complement what we learned in the Human Genome Project. And so, and after all, I think we'll unlock a really more holistic approach to disease prevention and even more effective and personalized interventions. So this is sort of our way of looking at the neuroexposome and noting that there are several other papers with slightly different figures, but look at them, they all have in common these kinds of risk factors. And here we just broke them up into three different areas. One is the exogenous, which is things like environmental toxicants, light, noise, and climate, of course. Endogenous factors like the microbiome and genes as well as part of the endogenous factors. And then the behavioral factors, of course, the psychosocial effects, but also things like lifestyle and sleep and stress. And yeah, this is a paper we did in your room a while back. So the NINDS neuroexposome strategic plan. So the goals for this, the first is just to better define what part of the exposome research is within our mission space at NINDS, and also which are the prioritized areas of neuroexposome research that we want to focus. We want to identify best practices, identify best practices for fostering more collaboration, which is absolutely imperative if we're going to do anything, is to get more collaboration, and identify tools and resources that we need to do this. And there's two strategic planning work groups, the work group of NINDS Council that identifies research priorities, and then an internal NINDS strategic plan development community to develop a implementation plan for these priorities. So yes, I mentioned that the council working group includes top of the line expert scientists in the field with Dr. Pam Lyon as our chair. The charge for this group is one, to develop the research priorities for NINDS, and then secondly, recommendations on bringing together team science with neuroscientists and the best tools and resources that we need. Both of these were discussed in separate workshops a while back. So finally, we are very, very interested in the public engagement. Early, we had a request for information on interdisciplinary research. As I mentioned, 2 mini workshops on team science and tools, and today's open session where we hope to get comments or questions from you all. And we also have another RFI for more input that is due by, July 31st. So that's it, and I will turn this over now to Drs. Pam Lyon and Gary. Thank you.
Thank you. Gary, I think you lead off with the next 2 slides.
Okay, let's see that next slide. So starting on this is really thinking about the definition of the exposome. And while Chris Wilde coined this term almost 20 years ago, there's been a lot of debate and various interpretations about what we mean by this. And so when we're talking about the neural exposome, we first have to understand the exposome. And so in one of the efforts to really harmonize around a definition, a meeting was convened at the Banbury Center of Cold Spring Harbor Laboratory back in December that brought together about 25 people from all over the US to dig into this and really come up with a consensus level definition of the exposome. And this is what has come out from this. We have a meeting report available. We have a manuscript that describes how we got to this point and this definition that is under review for publication. Hopefully we'll get that out in the next few weeks. But the idea is the exposome is this integrated compilation of all physical, chemical, biological, and psychosocial influences that impact biology. And when you look at it from a field standpoint, looking at exposomics, it's that field that studies that. And it's a comprehensive cumulative effect. So we're talking about throughout the lifespan and how these impact biological systems. And a big goal of this is really to focus on how we discover new things and how environmental factors influence that. And so the hope is that this definition becomes widely adopted. Many colleagues in Europe have pretty much adopted the same definition framework within the European Commission, and we're hoping to kind of harmonize around that. So next slide.
Oops. 1 second, it's not moving. There you go.
So with that, we can just think the very easy derivative of the exposome, when we think about the neural exposome, is that biological system we're studying is the nervous system. And so when you think about how all these complex exposures affect our brain function, peripheral nervous system function, the nervous system. And so the neural exposome is just having this focus on the nervous system. And so when we think about this throughout, again, we're talking about the physical, which could be noise and light and sleep patterns. It can be chemicals like pesticides in our environment or plasticizers or medications that we take. Biological, it can be infectious agents. It can be various fungal exposure or mold exposure. As well as social influences. So thinking about our social environment and socioeconomic factors, all the things that kind of can have this impact on our nervous system. And so we use this working definition throughout this, And we think it's very important to keep on this consistent definition to avoid confusion in the field as we go forward. So this is our working definition as we talk about the neural exposome, and hopefully this is something that becomes widely adopted. So Pam, back to you.
Thank you. Next slide, please. So I'm going to preface this slide with a little bit of background for the audience. This working group was called together several months back and charged with the 3 articles that David outlined for you in an earlier slide. And one of the things that I think has made this a very interesting charge and a rather complex charge to deal with is that when we think about the nervous system, it really is the system in our biological body that integrates all inputs coming into the biological organism. And so there's a lot of things that can influence nervous system function. This concept of the neural exposome is a very broad-reaching, very complex, all-encompassing definition of all the things that can impinge on the nervous system. And so trying to really bring this together into a set of priorities, definable priorities that are within the mission space of NINDS was a challenge. And so therefore, we're really looking forward to the public feedback on what we have as our draft priorities, because it has been an interesting and challenging exercise to try and pull together priorities that would be implementable. One thing that came out of our discussions, and I should mention that we broke the working group up into 3 subgroups. Each subgroup went off and sort of addressed the same set of questions, and then we came back and identified where we had areas of overlap, where we didn't have areas of overlap, what were the reasons for that, and did it inform the rest of the group. And so one thing that we kept coming back to over and over and over again in the context of these subgroups coming together in the large working group is that all of the subgroups kept talking about the fact that we really needed to shift research within NINDS mission space from focusing on single environmental exposures to research that really assess the composite effect of multiple exposures on multiple biologic targets. So that is sort of an overlying theme that came across all the various different components that the work subgroups are bringing back to the large working group. And again, I think this reflects the fact that the nervous system is an integrating system and is being influenced by all the environmental influences that are impinging on the biological system, including those that are endogenous as well as exogenous. And so again, this is sort of an overarching principle that is really relevant to all the particular draft priorities that we'll get into in the next couple of slides. The other overarching principle is that if we really thought about the NINDS mission space and really what is NINDS excelled at in terms of the NIH portfolio, research portfolio, and that has really been their emphasis and leadership on fundamental neuroscience to really understand the mechanisms underlying neurological disease, the biological mechanisms. And therefore, another overarching principle that came out across all the subgroup discussions is that really NINDS should leverage this global leadership in fundamental neuroscience to advance mechanistic research to really understand how the exposome influences neurological health and resilience, which is another factor we kept coming back to over and over again. It's important not only to understand how environmental exposures are promoting disease, but also how are environmental factors either promoting or decreasing the natural resilience of the nervous system to change. And then the third overarching principle was, if we looked across sort of this large sector of different environmental exposures, which you've heard both Dave and Gary talk to you about now, it's that we really thought that climate change was an environmental exposure that was urgent to understand, given its outsized impact on human populations and the world. And 2, that climate change really encompassed every one of those sectors of the exposome. We know that climate change can impact external factors, whether it's exposure to environmental pollutants. It can affect internal factors. What are the effects of temperature on internal homeostatic mechanisms? It affects behavior. We've seen this repeatedly in the human population when we have an extreme climate event, that there's great mental stress that's associated with that and the humans that are impacted by that climate change. And then, of course, social stressors are another very large part of climate change. So this seemed to us to be sort of an overarching factor that we really needed to grapple with, and the NINDS might be really well positioned to really challenge this, take this on as a challenge, and figure out what are the mechanisms by which climate change is driving changes in the exposome and how is that affecting neurological health. Okay, next slide, please. So within that broad umbrella of those three sort of areas where we think there's a great urgency if we're really going to advance our understanding of the neural exposome, we came up with 6 priority areas, and I'm calling them areas. The first one was really to address or identify key data gaps in the neural exposome. The second was to really focus on mechanistic research and what types of mechanisms would be important to understand to advance neural exposome research. Third was to really think about it from a public health, clinical, and interventional research perspective. So how can we take this mechanistic information and really use it to inform public health approaches, clinical approaches, et cetera? The fourth were what type of research resources and tools do we need to advance the neural exposome? Fifth, what about the research workforce, which is a type of resource, of course, but deserved a sort of special attention, because I think one of the challenges is really generating the workforce that has the knowledge that's broad enough to understand both environmental science as well as neurological science. And then the sixth is collaborations and partnerships. This is a huge problem. It's not going to be solved by NINDS alone. It's going to require partnerships across different agencies, across different programs, both within NIH and outside of NIH, to really make any significant advances in understanding the neural exposome. Next slide, please. So I'm going to take each one of those priorities and give you a bit more detail on them and where the working group is thinking at this moment. So in terms of priority research areas to address key data gaps, of course, we've already talked about the climate change as sort of being an overarching principle. But in the context of priority research areas, there's a real need to emphasize understanding the interactive effects of multiple exposures on multiple biological targets and pathways. Our understanding of the nervous system has advanced to the point we do have a great deal of insight on the impact of multiple organ systems on nervous system function. And so when we look at these exposomic factors that are influencing neurological health, it will be important to consider how they're affecting other organ systems or other biological targets and how those integrate to change neurological function. And so we'll need multi-factor research studies, and we'll need to consider the composite effects of multiple exposure on human health across the life course. There's also, as we had previously alluded to, but to put a little more detail into it, a need to understand and address the neurological effects of climate change. So this includes things like extreme temperature swings and weather events. We heard some beautiful arguments from one of the working group members, Eve Martyr, and how changes in temperature and extreme swings in temperature can affect physiologic function and homeostatic responses, including in the nervous system. There's also much detail or much information out there how geographic and social disruption and displacement due to climate change can affect neurological function. And then there are a number of indirect effects of climate change on the nervous system that we're just beginning to appreciate, and where a lot more information is really needed to understand it and put it together into exposomic understanding of the nervous system. Next slide, please. Also, in terms of research areas to address key data gaps, we know quite a bit about known and emerging neurotoxic contaminants and how they influence nervous system function and development, but there's still a lot that remains to be discovered. And these are kind of interesting questions to consider, because from a public policy perspective, if we can identify specific contaminants that are changing neurologic function, resilience, then we have grounds for changing public policy, which can affect a broader swath of the population than individual targeted interventions or lifestyle changes. So pollution, including air, water, and ground pollution, was considered a priority area. Pesticides and persistent organic pollutants, many of which are known to change nervous system function, but have not really been explicitly studied in the context of mechanisms by which they promote specific neurological diseases, was another priority area. And then there's an emerging science regarding plastics, both microplastics and nanoplastics, which are prevalent in our environment and are known to access the human biological system, but we really yet don't have a really firm understanding of their effects on the nervous system. There's also a need to leverage sociobiological research, which has been funded for many years across many NIH institutes to promote brain health equity. So how do these factors interact with chemical exposures and with each other to change brain health? If we can leverage that research that's already available to bring it into the specter, or I shouldn't say specter, the sector of the neural exposome, that would allow us to develop a neurocentric social exposome, social determinants of health toolkit that many investigators could use to assess social exposome measurements instruments. We could also leverage other NIH efforts focused on behavioral and social factors to integrate impacts of external and internal factors on neurological outcomes. And then this sociobiological research could be used to focus research to better define how the life course exposome contributes to disparities at the individual and community levels. Next slide, please. With respect to mechanistic research, as you heard me state earlier, the committee really felt strongly that NINDS should leverage its leadership in fundamental neuroscience to advance mechanistic research and identify mitigation targets. So one suggestion was to define the biological underpinnings of neurologic resiliency across the life course and to elucidate how environmental factors promote or undermine neurological resilience. Second, to identify mechanisms by which diseases in other organ systems, especially the cardiovascular, hepatic, pulmonary, and epithelial systems, influence neurological diseases. Next slide, please. Continuing under mechanistic research, another priority area was to promote research on how diverse environmental factors interact to promote neurological disease. This could include examining interactive effects between social and behavioral factors with external and internal factors, research on complex mixtures of chemicals. As I said, we have a pretty good database on how individual chemicals affect the nervous system, although we still need research on understanding how they're doing that. But we really have very little research on how the complex mixtures of chemicals to which we know humans are exposed is affecting the developing and mature nervous systems. We also need to identify mechanisms by which multiple environmental stressors interact with other omic factors, such as the genome, the epigenome, and the microbiome, as well as the transcriptome and metabolome, to influence individual risk of neurological disease. So in other words, how do these external and sociobiological factors interact with all these endogenous factors that we know are very important in neurological disease and resilience? Next slide, please. Then with respect to the priority to focus on public health, clinical, and interventional research, some of the sort of sub-priorities that were identified were first to identify exposome risk factors and neurological diseases in diverse human cohorts. Much of the information that we have has been generated in a very homogeneous population, which we know is not representative of the global human population, let alone the United States population. And so we really need to look at these factors in diverse human cohorts, understanding exposome genome and exposome epigenome interactions across diverse individuals. In addition, there's a need to assess the impact on neurological health of policy and community interventions that modify exposomic factors. We're seeing quite a bit of movement across different levels of our community, whether at the state or at the municipal level, government level, et cetera, and across the world, that are attempting to modify exposomic factors, but we don't really understand the impact of those modifications on neurological health and resilience. So there's a great need for those types of studies. And also sort of taking advantage of these policy and community interventions is sort of a natural experiment, if you will, that NINDS might well think about positioning itself to leverage to really understand how changes in exposomic exposures can really change the brain or brain health. Third would be to develop and test strategies to mitigate exposures, including studies to better understand, assess, and expand resilience. Next slide, please. The fourth priority area, and there's a lot of slides on this one, was to really discuss what types of research resources and tools are needed to advance neural exposome research. And we broke these down into sort of three categories: biorepositories and data repositories, infrastructure, and new model systems. Next slide, please. With respect to biorepositories and data repositories, these are seen as key to really advancing understanding of the neural exposome in human populations. In order to optimally utilize and develop these biorepositories and data repositories, the committee felt it would be necessary to develop validation and harmonization of exposome assays, so you could actually compare data across different cohorts. And this included the exposome assays, the metrics and measurement approaches across the life course. It would also be important to develop assays and exposure models to measure current and prior exposure to toxicants. There's a lot of effort going on in this area right now to, for example, take teeth and hair that's been archived to determine what prior exposures could be in any given cohort. More research along those lines is really needed to develop a very detailed understanding of the neural exposome. In addition, it would be great to better define biosamples and measures that could be uniformly collected across cohorts. So again, we would have some common metrics to compare results across diverse populations. Fourth would be to develop resources and approaches for collecting and analyzing life course geospatial data. So there is quite a bit of geospatial data that has been collected for a number of reasons by a number of agencies over the years. And being able to really tap into that or leverage that information to get a better sense of a specific community or specific individuals' exposures over their lifetime would be essential for advancing neural exposome research. Another goal of the working group was to develop templates or guidelines for integrating relatively low-cost add-on collection of biospecimens, and exposome variables. For example, there are a number of cohorts already ongoing that are looking at neurological health outcomes. It would be awesome if the NINDS could develop templates or guidelines for those investigators to add on from some very simple measures to collect information that would be important to assess the neural exposome. One example that was brought up by some of the working group members is just collecting data on occupation of the individuals over their lifetime, and also looking at maybe what was the environmental conditions on the day they report to a hospital, for example, for medical care. The last was that there's an urgent need, and this is really truly an urgent need, for integrative pathway analysis for assessing the impact of multiple environmental perturbations on an individual's nervous system. In other words, how do we harness machine learning and artificial intelligence to really integrate these very diverse data streams that are being generated, most of which consist of what we would consider to be very big data or high content data. Next slide, please. There we go. Thank you. In terms of research resources and tools, another area was infrastructure, which kind of follows on that last bullet in the previous slide. So there really is a need to develop guidelines and invest in tools for sample collection, storage, and analysis, including harmonization of protocols for laboratory and data analyses. I think the working group all agreed that in order to significantly advance the neural exposome, we're going to have to look at data being generated by multiple studies across multiple different cohorts. In order to enable a meaningful assessment of all those diverse data streams, we really need to develop these tools for harmonizing how samples are collected, stored, and analyzed. In addition, there's a need to develop guidelines for neural exposome research that address ELSI considerations related to personal protections, privacy, community trust in science, etc. Third would be the need to develop secure data management platforms that enable linking of a variety of data. And then fourth, developing robust, diverse clinical registries to generate a large pool of patients from which to recruit for prospective neural exposome studies. And again, a particular need in developing these registries would be to ensure the inclusion of diverse populations to allow for varied genetic backgrounds and cultural backgrounds and sampling of assorted biomes. Next slide, please. And then finally, under research resources and tools was the need to develop new model systems. So because we're looking at interactive factors here. So looking at the combination of multiple chemicals, of chemicals, sociobiological factors, et cetera, there really is a need to design novel methodologies to screen environmental stressors, to identify those that converge on mechanistic pathways that we know underlie the pathogenesis of diverse neurological diseases. This would inform then the epidemiological and field studies that are being done in human cohorts. And would also assist with mechanistic understanding of these interactions. There's also a need, or perhaps an opportunity, to leverage spontaneous neurological diseases in non-human models that are comparable to the human neurological diseases. For example, many companion animals are now known to develop neurological diseases very similar to those seen in their human owners. For example, epilepsy, Alzheimer's disease related dementia, and they're exposed to the same environment in many cases as the humans are. And so because their life spans are much shorter than a human, it may be more readily accessible to determine which exposomic factors are driving neurological disease and or resilience in those non-human models. Next slide, please. And then another priority area was developing a research workforce that was equipped and trained to really conduct really rigorous neural exposome research. So there's a need to train and educate people to really engage in this type of research. One approach for doing that, what we thought was within the mission space of NINDS, was to establish graduate and postdoc training grants on multidisciplinary exposome research. Another was to develop educational opportunities for investigators at all levels of training, but especially at the early stage, to broaden their skill set and orientation to examine the role of the environment in brain health. One of the things that the committee found in some of our investigations is that many neurologists really don't have an understanding of or appreciation of the neural exposome. And in order to engage those investigators who have the know-how in neurological disease in exposome research, it would be important to give them the training to understand exposome and how it impacts neurological disease and resiliency. It would also perhaps be an opportunity to recognize outstanding educators in neural exposomics and to leverage their expertise and experience. Next slide, please. Another approach for building the research workforce that would be needed to really conduct neural exposome research would be to cultivate interdisciplinary research teams and team science research approaches. One method that NINDS might consider for doing so would be to attract new talent by supporting neural exposome projects that deliberately require different disciplines to work together. NINDS has initiated such an approach, and I think it's been fairly effective so far, which is to require that any R01 being submitted integrate a neurologist and an environmental health science expert as a multi-PI team. Second would be to support the integration of exposomic expertise into ongoing human cohort studies. Could there be supplemental funds that NINDS could set up to really support ongoing cohorts to recruit individuals with expertise in the neural exposome to aid in their studies? Third would be to ensure that review panels have the appropriate interdisciplinary expertise to evaluate exposome-focused research, including exposome-wide association studies. Another area for NINDS to consider would be to communicate the importance of advancing neural exposome research. A relatively low-hanging fruit would be for the NINDS to update their website and other communication materials, including the mission statement, to convey their commitment to fostering neural exposomics. Another would be to educate the public, policymakers, clinicians, researchers, disease communities, and journalists in the role the environment plays in brain health. And I think they've started along this, for example, by the publication of their report in Neuron, but clearly there's more room for this. Next slide, please. And then finally, given the breadth and complexity of the neural exposome research arena, it was really important to the work group to recommend that the NINDS really foster institutional collaborations and partnerships. This is a big problem. It's really unrealistic to expect the NINDS to solve this on their own, but they should leverage their expertise along with the expertise of other agencies, complementary expertise of other agencies to promote neural exposome research. This would include seeking opportunities to expand existing human cohorts to include measures of neurological health. For example, and these are just examples provided by the working group, they're not meant to be inclusive. There's certainly many other examples, but one would be the NIH All of Us program. Can the NINDS interact with that program to basically integrate measures of neurological health into that existing program? Another one would be NIEHS's Human Health Exposure Analysis Resources, better known as HERE, and you can see a slide for that to the right. In addition, the working group would recommend that the NINDS seek input and advice from other NIH institutes and other agencies, many of which are instituting or initiating exposomic research programs of their own, and see how they can basically leverage each other's resources and expertise to broaden the playing field. Third would be to offer supplements to awards administered by NINDS and other NIH institutes that focus on neurologic health to integrate measures of the exposome. And then fourth, to support cross-institute research projects to fund neural exposome research that leverages the experience and mission of the NINDS and the partner institute. Next slide, please. Along these lines, thinking outside of the NIH, I guess the next one is still within the NIH, but the NINDS might consider collaborating with NCAT's CTSA program to incentivize clinical research on the role of environmental exposures on the progression and expression of neurological disease, but also to build partnership with government agencies that currently track and or set standards for environmental quality. So this might include government agencies such as the US EPA, and others, NIOSH, I say not NIOSH, but NOAA and other types of agencies that are collecting this type of data. Then engaging in interagency cross-sector efforts to establish a national or perhaps even global toxicant registry with exposure to air pollution, herbicides, pesticides, nanoplastics, etc., that are mapped to specific geographic units and a national global monitoring program. Next slide, please. So that is sort of an overview of the draft priorities. It's a lot, I know, and we would really appreciate feedback from the public as to areas that we have either overlooked that you think are very important, or how we may refine and focus the priority areas to the NINDS. And so thank you for your attention, and we're now open for questions from the public.
And Dave, please go one more slide. Thank you so much, Dr. Line, and I want to thank the entire NEXT working group who have dedicated a lot of time already in discussion and service of the neuroscience research community and really the American public broadly. My name is Sarah Dodson. I'm on the NINDS team that is supporting this planning effort. I would like to formally open the meeting up now for any public comment and discussion up until the end of the hour. So that gives us around 20 minutes or so. The working group will take all comments into consideration as they draft their final report over the next few weeks. As mentioned earlier, and I see we have one comment in the Q&A box already, but there are two ways to provide input. Please do use the Q&A box. I ask that you make those comments or questions as concise as you can, and hopefully we can get through everybody's input before the end of the hour. For more extensive comments, definitely encourage you to utilize the RFI, the request for information, that you can find in the web link that is listed here on this slide. And at that link, you can also access a PDF copy of the entire text that Dr. Line just walked through and description of those draft priorities. So now I'm going to go ahead and turn to the Q&A box. As I mentioned, I have seen one entered in here. So from Mark Scher, and I'm just going to read this out loud. I'm a pediatric neurologist focused on fetal neonatal neurology practice, education, and research. Would like to offer brief comments advocating for a life course approach that is inclusive of reproductive and pregnancy, including the placenta, as it influences the dynamic neural exposome across the lifespan. 3 critical windows during which neuroplasticity changes have critical sensitive effects based on gene-environment interactions. The first 1,000 days, adolescence, and reproductive senescence. Addressing health disparities is the paramount challenge to offer healthcare equity with interventions that integrate prevention, rescue, and reparative strategies. Advocacy must emphasize women and children through DEI who will later experience toxic stressor interplay across the lifespan with transgenerational effects and consequences. Thank you. Pam, any further comment on Mark Scher's input?
Thank you, Dr. Scheer. Yes, I believe the working group would concur with your recommendations. Looking at exposures across the life course has been a key element in all of our discussions and will feature broadly and strongly in the final recommendations that we put forward. But I thank you very much for emphasizing the importance of considering life course exposures. I will call briefly on Bob and then maybe Amy, if you have anything additional you'd like to add to that.
Nothing that you also didn't also say. We are looking at a life course approach that whether or not you're exposed, the dose is perhaps in some cases not as important as the timing of the exposure. So being exposed in pregnancy or early life may actually meet at a time when you're more sensitive, may actually be more toxic than a similar or even higher dose at other life stages. And the same may be true for elderly populations. So that's an excellent point.
Thank you, Bob. Amy, anything to add?
Yeah, I would absolutely concur. This issue of critical windows of exposure is something that is top of mind. For the work group, it was something that we absolutely discussed. So I'm so grateful that this participant has brought this comment forward. In addition, we have taken a very holistic view of life course, thinking of multi-generational exposures as well as intrauterine exposures. So thank you again.
Thank you. Sarah, back to you.
Okay, excellent. We have a second comment that has just come in. Again, I'm going to read it. This is from Amar Dand. I am a physician social scientist who works on the effects of the social environment on brain health. One comment. One second, it jumped for me. One comment I have is the conceptual definition of mechanism. Mechanism need not only be cellular biological. Mechanisms can be social, physical, chemical, or psychological that respects the breadth of factors you aim to study. For example, Snow discovered that the mechanism of cholera epidemic, which affects the brain, was a water pump, and removing its handle is the treatment. To foster interdisciplinary teams, a more open understanding of mechanism would encourage other specialists to engage. Has this line of thinking been considered? Pam.
Yes, thank you very much. We have absolutely considered that line of thinking. So we have a couple of experts on the working group that really specialize in that type of research. And very, very strongly, we are considering how social factors are influencing neurological health, brain health, and also how do these sociological factors interact or interface with other exposome factors. So, for example, chemicals to influence the neurological health. So we are considering that. I appreciate you emphasizing that for us. It is a very important consideration in our priorities. Let's see, I see that I have both Amy and Gina on here, and they are sort of our resident experts in this area. So Amy and then Gina, anything additional you'd like to add?
I think you covered it very well, Pam. This is an area that truly is at the cutting edge. So thank you to Dr. Don for bringing this forward and to emphasize the importance of not just siloing this research in any one of these domains, social, physical, chemical, or psychological, but the need for cross-cutting, multidimensional research that looks at interactions across all of these elements and how they interplay to ultimately promote health or to create disease. So thank you very much.
Thank you, Amy. Gina, anything?
Yeah, I would just say that it's absolutely critical how people respond to various exposures could be definitely affected by, for example, their social environment. And one study showed that loneliness imbues a 17% risk of dying for elderly people compared to those who are well integrated. So it is a huge factor. And I'm so glad that Amardand has brought it up.
Thank you. Sarah?
Thank you. One more comment here from Peter Spencer. FYI, a similar range of exposomic factors has been adopted by the World Federation of Neurology Environmental Neurology Specialty Group, for which I currently serve as the chair. A global approach to the subject includes attention to natural as well as synthetic chemicals and neurotoxic potential in normal and abnormal nutritional states. For example, environmentally tolerant plants like cassava and grass pea, i.e. complex chemical mixtures, are major sources of food on the African continent, and both induce pockets of highly prevalent irreversible neurological disease. The ENSG incorporates the principles of One Health and developmental origins of health and disease, and he thanks the working group.
Thank you, Peter. Nice to hear from you. Peter and I go back a ways. Yes, the working group would totally concur with your comments. We are certainly looking at multiple exposures as being key to understanding the neural exposome, and this includes multiple chemical exposures, but also diet. You bring up a very important point that nutrition and diet are also very important in determining how the brain responds to any type of exposomic factor. So we do have a couple of other neurotoxicology experts on the line here, so I'll turn this over to Gary and then to Ray, if there's anything you would like to add.
Yeah, I'll just comment about the global aspect of it. Yeah, we totally agree with this. I think this is something that, while NIH, tends to have this U.S.-centric aspect, they fund work around the globe. And I think it is important for us to think about how different exposures in different parts of the world, different social factors, different chemical exposures are very critical to it. And one of the long-term goals is to develop like a global exposome reference. Like, what are people normally exposed to in different parts of the world? But I agree. I think the work that ENSG is totally aligned, and I think it would be a very good partner as we move forward on things.
Ray?
Yeah, I agree completely. I think, taking a very concrete example, Parkinson's disease, I think in rural areas, I think pesticides are huge factors for contributing to Parkinson's disease. And in urban areas, for example, the United States, where rates of Parkinson's are higher in urban areas than rural areas, I think it's chemicals like trichloroethylene and perchloroethylene widely used in dry cleaning. And if you look at this globally, I get really concerned about sub-Saharan Africa. The use of pesticides has tripled there in the last 35 years, and 60% of the population's engaged in agricultural work, and the population's overwhelmingly young. To the earlier comments about concerns about early exposure to these toxins, I think that's where we should be focusing a lot of our efforts on.
Thank you, Ray. Sarah?
Sure. Next comment is from Latasha Crawford. In the interest of expanding the study of spontaneous disease in companion animals, which is an excellent idea, it will be important to ensure veterinarians, veterinary residents, and veterinary clinician scientists are included in your opportunities for training grants, supplements, and research teams. It will also be important that there is well-represented veterinary and comparative expertise included in review panels. Also, neuropathologists can play a quite useful role in addition to neurologists. And I see Gary has already entered an answer in the chat as well. But Pam, you want to respond to this?
Yeah, thank you. So I think the working group totally agrees with you, and that I think in many ways veterinarians have kind of led the charge on this, because the concept of One Health has been preeminent in veterinary circles for many years. And so again, a very actionable item for NINDS to consider would be to ensure that these RFAs and other types of programs to engage the community in neural exposome research are placed before the veterinary community for their engagement as well, and that they're actively recruited to the review committee. So thank you, very good suggestions. We do have a neuropathologist with us. So Tim, would you like to respond to that in any way?
I'm actually not a neuropathologist, but I think all of this is appropriate and on target, and I have nothing else to really add.
Thank you. Anybody else on the working group would like to respond to this? All right, thank you. Next question then, Sarah.
I actually Sorry, I just wanted to also bring up, as in response to this question and one of the prior ones, that one of the things that we're considering is sleep environment. There are lots of things that impact sleep, including diseases, but also physical things in the environment that impact sleep, such as companion animals. They can both help people sleep more deeply and more calmly, but also can disturb one's sleep. So, and that can contribute to disease progressions as well in ways that are only starting to be understood. So, this is something that we're considering as part of the call. Yeah.
Thank you, Gina.
Okay.
Sarah.
Sure. The next comment. Thank you from an anonymous attendee. Thank you for this very interesting presentation. In earlier slides, I got the impression that outcome measures of the exposome in terms of molecular outcomes were somewhat narrowly tailored to changes in gene expression and the epigenome. My question, therefore, is to what extent has the working group considered effects of the exposome on protein function that may not impact gene expression or epigenetic regulation, but nevertheless cause adverse neurological outcomes, which may or may not lead to genome effects?
And yes, this is certainly foremost in the conversations amongst the working group. So we will have to make sure our wording doesn't convey the idea that we are only looking at gene expression changes. We're not. Neuropathological behavior outcomes are equally important, as are changes at the level of the protein. And so I think when we think about mechanistic research, we're thinking quite broadly and not just at changes in gene expression. So I know we have a couple of folks in the working group that are online here that have a great deal of expertise in mechanistic research. Would any of you like to follow up on that comment?
I'll follow up. I think this is critically important. Any of the outcomes, whether it's cell biological or genetic, I think have the opportunity to provide new ways to mitigate toxicity or prevent toxicity.
Thank you, Tim. Anyone else? Okay, back to you, Sarah.
Thanks. Jason Richardson has submitted the next comment. Great comment, Ray. I would add that in urban environments, there's a large amount of household pesticides used. In public housing, there is typically pest control, but many individuals go out and use pesticides on their own. Many studies have also shown that pesticides used in other countries can often be readily available in communities and have resulted in poisoning of children and individuals.
Ray, would you like to respond?
I agree completely. You don't need to convince me of the dangers of pesticides and their use in people's homes, schools, yards, farms, golf courses, you name it. I'm concerned.
Yeah, thanks, Jason. I think this is, you guys have summarized very well why it is that pesticides were called out as a priority research area. Many pesticides are developed to target the nervous system of the intended species, but they cross-react with the human nervous system as well. And we have a rich understanding of how pesticides affect the nervous system, yet we still don't really understand the mechanisms by which many of these pesticides interact with other factors or even on their own to promote specific neurological diseases. So a key priority area called out by the working group. Thank you for the comment.
I would also add that an exposome approach would also include things like differences in the social environment between rural and urban communities, or even the biological environments, such as zoonoses versus other types of infections. So an exposome approach would be much more holistic in trying to understand those differences.
Thank you, Bob. Sarah.
Mark Scher has another comment. Consider for theme number 6, partnerships with NIH studies such as HEAL, ABCD, and the Human Placenta Project. Philanthropy partnering with NIH such as the Allen Institute and Melinda Gates Foundation, and the WHO focused on the 17 Sustainable Development Goals, particularly through the Global South.
Thank you. That's a great comment. Appreciate it. Sarah?
I think we have one more right now. Let me move it over into the queue. From an anonymous attendee, a recent paper offers a strategy to research and discover factors underlying the etiology of long latency neurodegenerative diseases, early onset, conjugal, twin discordant, and clusters of sporadic ALS, pathway to discovery of etiology via lifetime exposome research, and provides a PubMed link.
Thank you for that. That's very helpful. I don't know if anybody in the working group would like to respond to that, if you're familiar with this paper. We appreciate you pointing it out to us. We will definitely take it into consideration as we move forward with the final draft of the priorities. Thank you.
We are coming close to time. I do want to pause just for another minute, just to make sure if there are any last questions that the public would like to, any of the participants would like to enter into the box, please do that now. Again, give it just a few more seconds just to make sure. Okay.
Well, thank
All
you,
right, well,
everyone.
perfect.
Your
Yeah.
comments are much appreciated. We'll definitely take them into consideration as we put together the final draft or the final list of priorities to the NINDS. So thank you again.